Again, again, Mona, again.
धन मेरी क्लास चल रही है.
Huh?
चुप चुप चुप. So, good morning, good evening, class. Welcome to the next session of your EduFabrica MBBT course. I was just actually working for my college at the moment. We have given so much task in each single day, and I lost my file somehow, and I'm very sad about that. That how could I lost my file? I want to recover this file, but I'm not able to do so. Yes. I think I have recovered it. Yes. Great, great. So I can be now free from that thing. So good evening, everyone. Um, so very warm welcome for today's session. So today, from today onwards, on only today, maybe tomorrow, it's mainly the theory work. We will study about various friction enzymes, your markers, DNA markers, and DNA fingerprinting. So these three things will be covered in this topic today. Hello. So MBBT, DNA-based markers, RFLP, AFLP, RAPD, SSRs, MAS. So DNA markers includes your molecular markers, basic principle of molecular markers detection, molecular markers based on DNA hybridizations. Then we will discuss based on your PCR amplifications, random amplified polymorphic DNA, amplified fragment length polymorphism AFLP, then MAS, molecular marker assisted selection. So in the case of um, molecular marker, which is a DNA sequence in the genome which can be located and identified. So they are the one by which we can do alternation, like mutations, insertions, deletions, based on the particular compositions of genomes in different plants, in different, and further they're collectively called as polymorphism, which can be mapped and identified. On top, plant breeders always refers to detect these genes because they are very highly valuable, because once you find your molecular marker, let's say for drought tolerance, you can find somewhere nearby also for the salt tolerance, something for the high yield. So all these few markers could be very helpful in order to give us a very good variety of plant species. So in that case, they are very helpful, very useful. So they are quite reliable and advantages in plant breeding programs, and they provide two representations of genetic makeup at the DNA level, and they are consistent, not affected by environmental factors. And molecular markers can be detected much before the development of plants occur. A large number of markers can be generated as per the needs. So basic principle of molecular marker detection. So how do you identify them? Basically, let's say you have two plants of same species, one with disease resistive, one with the other disease resistance. And then what you do is you extract the genome from that DNA, or you extract the DNA from that genome and digest with the restriction enzyme and separate it by gel electrophoresis. The DNA fragment can be detected by separation, for instance, disease-resistant plants or may have shorter DNA fragments, while the disease-sensitive plants may have longer DNA fragments. So I will show you this in the next figure. Whereas the molecular markers are of two types, that is DNA hybridizations and based on the PCR amplifications. So based on the DNA hybridization, there could be non-PCR based approaches, or based on the PCR amplifications, there could be PCR based approaches. So molecular markers on the left and right hand side, what you're seeing here is a disease sensitive plant and disease resistance plant. So disease sensitive plant is like we can extract DNA from it and from both part of it, and then we digest them with the restriction enzymes or gel electrophoresis. Then the shorter DNA fragments could be seen from the disease sensitive plant, whereas the disease resistance plant will show us a longer DNA fragments. So that's the basic principle of identifying molecular markers detection and screening of genotypes for the identification of DNA markers. So molecular based on DNA hybridization, so the DNA piece can be cloned and allowed to hybridize with genomic DNA which can be detected. Further, marker based DNA hybridization is widely used, and the major limitations is required with the large quantities of DNA and use of radioactive label probes. So first, based on the DNA hybridization comes your RFLP, that is restriction fragment length polymorphism. So RFLP was the very first technology that were employed for the detection of polymorphism based on the DNA sequences at differences. Where RFLP is mainly is based on the alternative friction enzyme sites. So the outline will be shown in the next slide. So basically this also includes the same principle that we have discussed at the start of this lecture, that is isolation of genomic DNA, digestion of restriction enzymes, separation by electrophoresis, and finally hybridization by incubating with the clone and label probes. So that's an outline of RFLP. That is, you have isolation of genomic DNA, then you digest them with the restriction endonucleases. Then you have various small DNA pieces separated by agar gel, electrophoresis, and then DNA pieces transferred to a membrane filter, which we can incubate with the clone and label probes. And RFLP bands could be detected out of it. So based on the presence of section size, DNA fragments of different length can be generated by using different section enzymes. So in the figure 53.3 here, what we can see is two different plants. Plant A has mutations leading to the loss of the restriction sites that can be digested by the E. coli 1. So plant A will not be restricted by E. coli 1, but whereas plant B could be restricted by E. coli sites. So when we put them over the gel electrophoresis by adding the these restriction enzymes, so we can see on the left hand side is a monomorphic pattern, that when we add the restriction enzyme, mainly the hint 3, so we can see there are two base pairs, that is 200 base pairs and 800 base pairs for the both plant A and B. Whereas in the second case, the polymorphic pattern, there are plant A and B. In the plant A is around 1000 base pairs, where the plant B has 350 base pairs plus 650 base pairs. And this second pattern that we are seeing, this polymorphic pattern, is because it has the restriction sites and it does not mutate. So that's why we see this normal state of polymorphic DNA. Further, based on the, this is explained in this diagram with the plant source DNA, restriction, digestions, then electrophoretic separation, southern hybridizations, then the autoradiography. Then markers based on PCR amplification. So that was above was the non-PCR hybridized form with the RFLP. Now we are discussing the PCR based forms. That is your RAPD, RFLP, SSRs, SNPs. So basically all these other than RFLP part, others are based on your PCR base. And PCR we have discussed in our first lectures. That is polymerase chain reactions. We amplify one part. And within this PCR based markers, they divide into two parts: locus non-specific, locus specific. When it is locus non-specific, it's come under two different things, that is RAPD and AFLP. Whereas when it is in the specific markers, then it is in SSRs and SNPs. So let's start with the first one of RAPD, random amplified polymorphic DNA. So random amplified, as the name suggests, randomly we amplify this polymorphic DNA. So RAPD is a molecular marker based on PCR amplification. So here we can see in 53.4 in this figure, first we isolated the genomic DNA, then we denatured the DNA. But instead of in previous case, we were not having this annealing of the PCR. In this case, we are having these primers which will anneal to the DNA template, and the complementary standard synthesis will be produced. And then amplified products by gel electrophorese are identified. So single short oligonucleotide primers can be arbitrarily selected and used for amplifications DNA segments of the genome, and the amplified products are separated on the electrophoresis and are identified. So when the DNA molecules are digested by enzyme HINT3, there is no difference in the DNA fragments separated. However, with enzyme E. coli 1 plant A DNA molecule is not digested, while plant B DNA molecule is digested. So this results in polymorphic pattern of separation. So some of the applications, they could be used in the bean varieties, RAPD marker methods. So 12 samples were analyzed using 60 markers also. So here we can see that this thing of RAPD has produced 296 markers that could be scored, and almost 85% are similarly predicted. For example, DNA from plant A allows the amplification of sequence A, C, D, but it doesn't amplify plant B. So this indicates in plant A, primer sites for primer used not at the sequence B. So other sequences were sequenced with these primers, but not the sequence B was done. So similarly, a DNA sequence alteration at one primer binding site for sequence A has prevented from being amplified when DNA from plant 2 is used. So in plant A, we can see that when we put them for the amplification, then the sequences of A, C, D, A, C, D has been amplified, whereas the B was not. But in the case of second, your A and B both were not set. A and B were not amplified, whereas in the third and fourth, is the same case as the first one in the plant variety, where in the fourth one C and D were not identified. So that's how the main difference. Further, RAPD markers have been used to identify several disease resistance genes like RP94 genes, which is responsible for resistance to stem rust in barley, and they are identified to link to this gene. Further, some of the demerits that the constraints about reproducibility of the results and null alleles are not directly detected in them. So further, in the same case, based on the nucleotide alternation in the gen, the polymorphism in the genome, the polymorphism of amplified DNA sequence differ, which can be identified as bands on the gel electrophoresis. So next part is AFLP, that is amplified fragment length polymorphism. So here they are the not random one, but it's a novel technique which employs a combination of your first one, the RFLP and RAPD together. So AFLP includes the basically generation of a DNA fragments using friction enzymes and oligonucleotide adapters, and their amplification then we do by PCR. So first we restrict them with the friction enzymes, we digest them with the friction enzymes, then we do the PCR. And this technique combines the usefulness of both your friction digestion and PCR. So DNA of genome is extracted in this case, restricted with enzymes, mainly by two enzymes, that is MSC1 and frequent cutter, that is E. coli 1. The cut ends on both sides and then ligated to known sequence by the oligonucleotides. Then PCR is performed with the pre-selections of fragment of DNA, which has a single specific nucleotide. So further reduces the pool of DNA fragments to manageable level. An autoradiography can be performed for the detection of DNA fragments. And use of radio-labeled primers and fluorescently labeled fragments cook in this AFLP. So here we can see amplified fragment length polymorphism. On the left side, the diagram is showing there are two templates of DNA molecule from 5' to 3' end, 5 to 3' to 5' end, and we restrict them with the E. coli 1 and MSC1. And then what we are seeing is here is a 5' to 3' end, the DNA molecule is being restricted by showing this at the 3' end. After T, your AATT is being removed. Then we ligate with the linkers. Then we have these linkers as A, yeah. And also here it's remain like that. So our CTTA GGC remains same like this. Your GTA TTTA we remove this A and then we have this A here. And then we do the PCR employing the nucleotide extension. So CTTG and GGTA. So here was a single A and then they will amplify and they will go amplify like that. So that's how the amplified fragment length polymorphism is done. So this is very useful in genetic mapping. Uh, Several economical important cereal crops such as rice, barley, wheat have been mapped with the AFLP markers. And AFLP markers, which are produced by different combinations of reaction enzymes, are distributed throughout the genome. In barley, AFLP markers are located on the long and short arm of all 7 chromosomes. Further, length of polymorphism detected in barley by AFLP can range from 12.2% to 29.04%. And this breeding is for the resistance important program in cereal research also. Some of the merits that they are used to screen large number of polymorphism. It is possible to saturate specific regions of genome for map-based cloning of target genes. Some demerits, proprietary technique, null allele cannot be detected, required high amount of DNA, and some expensive techniques like silver staining, radio label is also being there. Then in the based on the second type, we have discussed so far the non-locus type in your RFLP and AFLP. Now we are discussing the locus specific type, that is your STRs and SSRs. So in STS, the sequence tag sites means they're uniquely simple copy segments of genomes whose DNA sequences are known and which can be amplified using PCRs. So STS markers are based on the polymorphism of simple nucleotide repeats. So like GATN, they will be repeating again and again CAA on the genome. So they have been recently reported in the plants. So when STS loci contains simple sequence length polymorphism SSLPs, they are highly valuable as a molecular markers, and STS loci have been analyzed and studied number of plant species. And the microsatellites, they are the tandemly repeated multiple copies of mono, di, tri, tetranucleotide motifs. So in some instances, the flanking sequence of repeat sequence may be unique. So primer can be designed for such flanking sequence to detect the sequence tag microsatellites. And this could be done by using PCR. And sequence characterized amplified region SCRs, they are the modified versions of STS. They are developed by PCR primers that are made for the ends of RAPD fragments. And SCRs are useful for rapid development of STS markers. This is SCRs. We can have amplified with primer of RAPD band and then amplified with a scar with the primer. So both are being, we can do these 2 different parts. Then come the third section of our study. So we have discussed since the lecture has been started that there is a non-PCR base, that is RAPD. Then we have discussed your RFLP and AFLP, that is your PCR based. And then they were non-locus. And under the non-locus then comes a locus, in which we have just discussed STRs, SSRs, microsatellites, SCRs. Now comes the third part of your studies in the molecular markers, that is your molecular marker assisted selection. So these are the desired traits and improvement of crop plants which have been part of conventional breeding programs. And this is predominantly based on the identifications of phenotypes. And what we do is that in the phenotypes don't necessarily represent the genotype. Many times environment which marks the genotype here. So following are the major requirements for molecular marker based in plant breeding, that they should be closely linked with the desired trait, quite highly efficient, reproducible, and easy to carry out. The analysis also should be economical in that sense. And then comes your linkage analysis. Linkage analysis basically deals with the studies to correlate the link between the molecular markers and desired trait. And this is important aspect of molecular breeding programs. And linkage analysis has to be carried out among populations of several generations to establish the appropriate linkage. So in the early years, linkage analysis was carried out by using isoenzymes, and associated by polymorphisms, molecular markers are now being used in that case. Then comes the quantitative trait loci, QTLs. These are many characteristics controlled by several genes in complex manner. Some good examples are growth habit, yield, adaptability to environment, and disease resistance. The locations on the chromosomes for these genes are regarded as a QTLs, and they improve in the complex character controlled by many genes. So they are the one which the golden rice has been developed with the provitamin A in them and involved the insertions of three genes that took about 7 years. So thank you very much. That was it, the DNA based markers. Bhai, what is this geodole? Yes, Saleh Edru. Come on. I think you should come and say your full name and talk to us. I have taken your screenshot um, just now. Who is this person? Please talk to me. My phone number is this one because I told already that today is the day that we will study. Oh man, this is very foul language. This is not acceptable. If you can, GI doll, if you have guts, please talk to me if you can. Otherwise, using this language that it shows that you are not a student. First of all, you are not here to study, and using this language. So we have done. I don't know. I don't want to defend myself here. You are some juggi wala or something, I don't know, who doesn't. What shall I say? Who? I mean, it's okay because today, as I already told, that the things will be taught from the theory based, and there will be virtual labs are not at the moment possible just because the Adobe Photoshop is not working. But beyond that, we are having very nice videos. I think that person doesn't even came to our classes. That's why using this absurd language and making the atmosphere of the class so bad. Even if I ask this person, could you tell me the various types of molecular markers that we have discussed today? Could you answer me? It's just that you are filled with a lot of hate, a lot of negativity, and a lot of, I don't know, pressure from other life of your life, which made these things to do students. If they are here to learn, and if I have to remove these, that you should have never ever used these languages here. I'm really hurt very badly. This is hate speech. Very sad, very sad. Can I see this person's community? Who is this person actually? Should I share this screenshot? I mean, the language that has been used today. Um, Come on. We are teaching like BTech, BSc, MSc. I mean, we are not here just. I'm really done. Okay, anyhow. Now comes the next part, that is your friction enzymes, which includes introduction, more of how they work, the start activity, the nomenclature of friction enzymes, type of friction enzymes, DNA ligase, kinase, reverse transcriptase, and clinofragments. So 1, 2, 3, 4, 1, 2, 3, 4, 5. Yeah, around 9 topics we will cover in this. So in this, like we have discussed the role of friction enzyme so far very well. In this RFLP markers, your AFLP markers, RAPD markers, that it help really to cut your DNA from the desired site. So let's discuss about its various types, how they are working. So basically, at present, no chemical methods can achieve the ability to manipulate the DNA in vitro. So only enzymes are able to carry out the functions of manipulating the DNA. So each enzyme has a vital role to play in the process of genetic engineering. So in the genetic engineering, following 7 enzymes are there, like nucleases, friction enzymes, DNA ligase, kinase, phosphatase, reverse transcriptase, terminal deoxynucleotide transferase, RNases. So let's discuss one by one. So nucleases are a group of enzymes which cleave or cut the genetic material. So they included 2 types, that is your DNAs and RNAs. So nucleases are further classified into 2 types based on the substrate. Nucleases which act on or cut the DNA are classified as DNases, and which cuts the act on RNA, they are known as RNases. So DNAs are further classified into 2 types on the position. So DNAs that act on the ends or terminal regions of DNA are called exonucleases, and those that act at a non-specific regions in the center of the DNA are called endonucleases. So exonucleases requires a DNA strand with at least 2 to 5 and 3 prime end, and they cannot act on DNA which is circular. Whereas endonucleases can act on the circular DNA and do not require any free DNA ends. So, and on the top, exonucleases release nucleotides like nucleic acid, sugar, and phosphate, whereas the endonucleases release short segments of DNA. Then comes the restriction enzymes. So DNA which act on specific positions or sequence on the DNA are called as restrictions endonucleases. And they are recognized. You're also called as restriction enzymes, recognition sequence, and these sequences are palindromic sequences. Yeah, palindromic sequences. No worries, Vishek and Vivek, but there is a way of teaching things, and there is a way of explaining things to students. Um, because I was also a student. Uh, I was also a student, and I used to have these classmates in my class that they just abruptly says very harsh words to teachers, and they just make the atmosphere of the class like I don't want good words or something, like want to appreciate myself. I'm in my own world somewhere of molecular biology, DNA, RNA, genomics, proteomics. I'm happy where I am. It's just I don't know. This is very hard time for all of us at the moment. So you never know what will be the next day. We will be living or not. My reason to be here is mainly is that um, that whatever the knowledge that I had in myself, that I love to share with my students or any my colleagues or any layman, my relatives, anyone. But it's always good to share things that we have in ourselves. It's never ever good that whatever the knowledge that we have gained in this past 16 years of biotechnology experience starting in 2005, if I keep them for myself for my only, um, that will be of no use then. So I love teaching. I like teaching. It's my passion. um, but these kinds of things sometimes makes fuzzy and yeah, anyhow. So we continue with the same pace here, that is your section enzymes. So we have discussed that they give this palindromic sequence like having ATAAGC, and if we read it from the left side or right side, they will be always similar. And different restriction enzymes present in different bacteria that can recognize on different or same restriction sites, but they will cut at two different points within the restriction sites. So these are called isomers, whereas where two restriction enzymes from single bacteria will cut at the same restriction sites. So what is the mode of action for restriction enzymes? It's a bit complex here, but let's have a read here that the restriction enzymes, first of all, they bind to the recognition sites and they look for the methylation site, that there is a methyl group present on that DNA. And then if it is present, then methylation will occur on the recognition site, that is your methyl group removed. Then it just falls off the DNA and does not cut. Then further it will not cut, it will just remain like that, your restriction enzymes. So if only one strand in the DNA molecule is methylated, only single strand is methylated, other strand is not methylated, then the only type 1 and type 3, these are called type 1 and type 3, will methylate the other strand required positions. So whereas in type 2 restriction enzymes, it take the help of another enzymes called methylase, and then it methylate the DNA. And then restriction enzymes clear the DNA. Further, if only by methylation mechanism, restriction enzymes are present in the bacteria, but they do not cleave the bacterial DNA, but cleaves the foreign DNA. That's the difference between eukaryote and prokaryotes. Then there is a star activity, that is sometimes restriction enzymes recognize and cleaves the DNA strands at the recognition sites with asymmetric palindromic sequence. So they are not like blunt ends. So BAM H1 cuts at the sequence BATCC. But under extreme conditions, such low ionic strength, it will cut or cleave on the following sequence of NGA-TCC, or like this one, or GNTCC. And when they cut the extreme conditions, not like even blunt end, so these conditions are called as star activity of restriction enzymes. And how we name them, how the naming culture of these restriction enzyme is done, it's basically a large number of restriction enzymes have been discovered so far, so it's very difficult to identify them and name them. So in that sense, Smith and Nathans in 1973 proposed the nomenclature namings. So every restriction enzymes could have specific names which would identify it uniquely. So what they did, they gave first three letters in italics, and they will come from the biological source of the enzyme. The first letter will be the initial of the genus, and the second will be second and third letter will be from the species name. If it is Escherichia coli, the first capital name will be E, and the rest from the species, the two letters from the species, CO. If it is Haemophilus influenza, then H from the Haemophilus, and then influenza IN. If it is Diplococcus pneumonia, then DPN, and so on. So then comes the next part, is the strain, that which strain of bacteria it is. Then it is strain R or which one. Then after the strain, If it is a first type or second type or third type, then we give them with the Roman numerals like this as the first enzyme or second or third. So E coli 1, E coli 2 like this. Same way we have different hin R1, 2, different DPN like that. So there are three types of friction enzymes we have discussed: type 1, 2, and 3. So type 1 and 3 have ATP dependent friction activity and modifications, and both of these are recognized unmethylated recognition sequences. So that doesn't require the methylated one, 1 and 3. And type 1 enzyme cleave the DNA at random sites, whereas a type 3 cleave at a specific site. On whereas the type 2 modification possess separate enzymes, that is your endonuclease and methylase activity, as we have discussed in the mode of action, and mostly widely used for genetic manipulations. So type 2 restriction enzyme is the most highly used, not the first and 3. So let's say in the type 1 restriction enzymes, these restriction enzymes recognize the recognition sites, but then they cleave somewhere between 400 base pairs to 10,000 base pairs or 10 kilo base pairs right or left. And cleavage is not specific, so and enzymes are made up of three peptides and they require magnesium, ATP, and acetylcyl methionine for the cleavage of enzymatic hydrolysis of DNA. And whereas the type 2 restriction enzymes, if we discuss, they are the one they recognize restriction sites and they cleave the DNA as where we want it, and they require magnesiums as a cofactor in cleavage activity, and they could generate 5' phosphate and 3' hydroxyl group. And they are of highly important because of their specificity. Further, based on their cutting, they could be of two types: blend end cutters and cohesive end cutters. So type 2 under type 2 restriction enzymes as a blend end cutters. So they are the one with enzymes of this class cut the DNA strands at the same points on the both strands of DNA. So within the recognition sequence. So they give mainly such like blunt end or flush end at the end. Whereas the type 2 cohesive ends, they are the one which gives different ends on the both sides of DNA within the recognition sequence. So they generate short single-stranded sequences at the end. And then type 3, the last one, that is the restriction enzymes that cut DNA at 1 kilo base pairs away from the restriction sites, and these enzymes are made up of two peptides or subunits. And these enzymes requires your ATP, magnesium, and acetylcyl methionine for the action. So that's a summary what we have just discussed just now. Type 1, 2, and 3. This is quite highly informational PPT here, that is for the students who are preparing for NET, GATE, or any external exam, GRF, or for the interview. So these questions are mostly asked the difference between three types of friction enzymes. So better prepare them. Yeah, bachcho. So your cofactors are like magnesium, ATP, SAM is there also for type 2, magnesium and SAM. For the type 3 is magnesium, ATP, SAM are required. Further types of restriction enzymes, so they could be 4 base cutters, 6 base cutters, and 8 base cutters. The 4 base cutters are the one like MBO1, DPN, SAO3AI. So they cut it, their recognition sites are GATC, so as in, and this is 6 base, they cut from the 6, and they're from the 8 base cutters like that. And then as we're discussing about the various enzymes that are used in the case of your DNA molecular modeling and the recombination DNA technology. So the next part that come here is your DNA ligase. Okay. So DNA ligase, the one which requires joining of your two different DNA segments. So two segments could be joined together, and the cohesive ends generated by some restriction enzymes will be annealed themselves by forming the hydrogen bonds. So any two bonds, any two DNA different segments they want to get joined in, so they could be joined with the help of your DNA ligase. And they could be covalently by catalyzing the formation of phosphodiester bonds between adjacent nucleotides. So DNA ligase that are isolated from E. coli and T4 bacteriophage is widely used. T4 ligase is the which requires ATP as a cofactor, and E. coli ligase requires NADP as a cofactor. So the cofactor is first split and then AMP binds to the enzyme to form your enzyme AMP complex. And for the better results, these ligase reactions are done at 40 degrees Celsius. Then comes your kinases, which is a group of enzymes which add your free pyrophosphate group to the any substrate like protein, DNA, and RNA, and they use ATP as a cofactor and adds phosphate by breaking the ATP into ADP and pyrophosphate. So very highly useful enzyme in the molecular biology and genetic engineering to add radiolabeled phosphatases. So there are some types of phosphatases here we will discuss now. So after the kinases, phosphatases, I mean, if you say they are the yin yang uh, of enzyme reactivity, these kinases and phosphatases. So if the kinases are adding the phosphate group, and on the other side, phosphatases work is to remove a phosphate group from the substrates of your DNA, RNA, and proteins. So they act upon the basic buffer around 8 or 9, and they are called as alkaline phosphatases. So most commonly are bacterial alkaline phosphatases, BAP, and also calf intestine alkaline phosphatases, CIAP. So they are mostly used in the molecular cloning experiments, and the PO4 from substrate is used by forming the pyrophosphorylated serine intermediate. So alkaline phosphatases are metalloenzymes and has zinc ions in them also. So let's discuss this BAP. And BAP has a dimer containing 6 zinc ions, yeah, and 2 of which are essential for the enzyme activity, and they are quite stable and do not get inactivated by heat or detergent. Whereas CHAP is also a dimer. It requires zinc and manganese for the action, and it is inactivated by heating at 700 degrees Celsius for 20 minutes or in the presence of 10 millimolar of EGTA. The next enzyme, reverse transcriptase enzymes, which is used to synthesize your DNA from RNA, the complementary copy. This is very highly useful, very special technique that is used for your production in the RT-PCR, your reverse transcriptase PCR. So those enzymes that are present in most of the RNA tumor viruses and retroviruses. And reverse transcriptase enzyme is also called as RNA-dependent DNA polymerase. Then comes your terminal deoxynucleotide transfer. So there are some another not so famous but still quite important enzymes types. So these are the one which polymerase adds nucleotides at 3' end. That is your clinio fragment and does not require any complementary sequence and does not copy any DNA sequences. Whereas there's a terminal deoxynucleotide transferase, TDNT, adds a nucleotide where it comes to its active site. It does not show any preference for any nucleotide. On the other hand, RNases are they cleaves your RNA at 5' end very specifically, and they complex enzyme consisting of small proteins, that is 20 kilodaltons and 377 nucleotide RNA molecule. So it has been observed that RNA molecule possesses at least part of the enzymatic activity of the complex. Hence, it is an example of your ribosome. Now, this is the last slide of our today's lecture. So we have discussed quite a number of things since morning. The lecture has been started. That is your various molecular markers based like RAPD, non-PCR based. Then we have discussed your PCR based markers, AFLP, RFLP, SSRs, microsatellites, and molecular based markers, QTLs, linkage analysis. Then afterwards we have today started with the various types of your enzymes and we particularly were interested with the restriction enzymes here, and we have discussed its three different types. And now we are going to discuss about your clino fragments. This is quite exceptional questions that come sometimes in interview or in your exams. That what are the clino fragments? So they are the one. So E. coli DNA polymerase, they consist of single polypeptide chain first of all. Yeah. So poly 1 carry out three enzymatic reactions that performed by three distinct functional domains. So two fragments are obtained from DNA polymerase, which is treated with a trypsin in mild conditions, and the larger fragment is known as clino fragments. So these fragments, they are made up of around 602 amino acids in the length, and the functions of clino fragment is to add nucleotide to the 3' end and exonuclease activity. So clino fragments add nucleotides by using complementary strands as a reference, and it cannot extend the DNA without the presence of the complementary strand. But if any nucleotide is added by mistake and the base pair is wrong in that, then they by using the 3,5 exonuclease activity present in the clino fragment and could be mispaired, using is removed. So that's it for today. I want to start with DNA fingerprinting today, but I think I cannot do it. So I will do it the next time tomorrow about that. We work on next time for the same thing. So I will share these restriction enzymes with you now. Oh, I have shared the wrong thing at the wrong place. Here I have to share. Okay, that's it for today. We see us tomorrow, same time, same place. Thank you very much. Screenshot.